Here's a paper that gives us fantastic news. The first being that with additional vaccinations, immunocompromised individuals can have the same level of immunity as healthy individuals. That vaccinating with different strains of the virus has done additive effect, where repetitive mRNA vaccine doses and/or these doses with Omicron infection caused a enhanced and diversified cellular response with minimal identifications of antigenic syn effect, which is when the virus that the body first encounters during an infection, the antigen that's produced for that first virus, is what ends up getting created for the new virus, even if this first antigen becomes recessive and a new antigen is predominant, which would make that initial immune response ineffective for the new variant. And we're not seeing this occurring, nor are we seeing T cell immunity functional exhaustion despite repetitive antigen exposure, with resultant solid immunological imprints, so the memory cells for ancestral COVID mRNA vaccination, where the findings of this study are highlighting the importance of booster vaccinations in immunocompromised individuals. Now let's dive into it. So at the beginning of the study, all participants had a negative PCR test with no previous documented COVID-19. However, when we look at the data, we see that there were some individuals that had a previous COVID infection. See here where the predominant is 0, but we still have different memory CD4 and CD8 T cells that are showing up in both our negative control and then D0, which is before they received the first vaccine. Now, patients in this study received 3 or 4 doses of the Wuhan 1-based mRNA vaccine, with samples collected before the first dose at day 0, before the third dose at 6 months, and then after booster vaccination at month 12. So the trend that we're seeing in both the CD4 and CD8 memory T cells is that more people are shifting from this original population over to having different levels of these natural immune cells, showing that the vaccination is working in a majority of people. The specific immunodeficiencies in the study include HIV, hematopoietic stem cell transplantation, solid organ transplantation, chronic lymphocytic leukemia, and then healthy controls. So at first we look at this and it's very overwhelming. So we'll break it down and we'll see at day 0 there is low CD8 and CD4 T cell memory, and then but still some existing because they had a prior infection that they didn't know about. And then it's increasing at 6 and 12 months in sort of a stepwise fashion, where at 6 months, especially for the blood transfusions and the solid organ transplants, there is very low conferred immunity. However, in all groups, by the time it went from 6 months to 12 months, they had approximately equivalent levels of immunity, where we're seeing the same trend with the CD8 memory cells. So this information tells us, one, that both individuals that have active immunosuppression in advanced age have a negative responsiveness to 2 vaccine doses. However, with additional vaccine doses, this goes away, and they have a pretty substantial vaccine dose that's equivalent to that in healthy individuals. And because the healthy individuals aren't like outperforming in memory cell immunity to these immunocompromised individuals at this 12-month follow-up, this is suggesting that there's sort of a ceiling where the cells become saturated, where you're at this optimal level. So vaccines work, especially if you're immunocompromised or even if you're elderly.